Procedure. In procedure, first of all, various apparatus: RBC fluid, that is known as Hems fluid, microscope, then hemocytometer box, which contains watch glass, then Nuba chamber, RBC pipette, and cover slip. Okay, so these are the instruments which will be used in this practical. Now you have to wash these instruments properly first of all before use, and then you have to wipe them with the clean handkerchief or white cloth. Then you have to use those instruments. Okay, now we will just collect the RBC fluid, that is Hems fluid, in watch glass. Okay, then we will focus Nuba chamber, empty Nuba chamber, under the 10x first of all. Put 10x, that is low power lens, into the position, then switch on the microscope. Now first of all, makes light adjustment first. In low power lens we will use least light, so intensity must be the lowest, condenser must be in the lowest position, and the iris diaphragm must be slightly open, so almost close. Okay, then make the position of the platform exactly underneath the lens. Okay, see from the sideways, Nuba chamber must be exactly under the lens, and from the front. Okay, after making sure, slowly make this platform downwards. Okay, now focus the central square exactly under the lens and change the objective lens from the 10x to 45x. Now again you have to change the light adjustment, and in 45x we will increase the light intensity slightly, condenser into the mid position, not lowest one, not uppermost, and this iris diaphragm must be partially open. Okay, after making sure, only you have to make fine adjustment and the focus will be there. Okay, so what you have to remember is making sure the light adjustment must be done before 45x focus. It must be in the mid level of the intensity, condenser must be in the mid position, and iris diaphragm partially open. Okay, after then you will be able to focus under the 45x. After focus, just switch off the microscope, pull out this Nuba chamber and put it over the platform. Okay, that's it. Okay, now about the pipetting. Now take the RBC pipette, then go for a prick with the help of lancet, collect the blood up to 0.5 marking in the pipette. After collecting the blood up to 0.5, wipe out the tip of the pipette with your finger, not with the help of handkerchief or cotton. Use your fingers for the wiping the tip of this pipette. After collecting blood, immediately collect the RBC fluid. Okay, now you have to collect the RBC fluid like this and make this pipette vertical while collecting, so that this bubble will come out of the upper end. Okay, so as you can see, the RBC fluid is filled up to the 101 mark. There should be no bubble inside the bulb. Okay, so make sure for that you have to make this pipette vertical. After collecting the RBC fluid, make this pipette horizontal, because in this pipette it is open at both the ends, from the upper end or lower end. So you have to make sure that this pipette should remain horizontal position. And you have to mix it for 1 to 2 minutes. While mixing, you can mix either by blocking these two ends like this, or you can mix it while putting in horizontal position between two palms like this one. Okay, so either of these two methods you can use either one. Okay, so like this you have to mix for 1.5 to 2 minutes. After mixing, put this pipette over the platform in horizontal position. Okay, it should not be inclined towards the one end, otherwise this fluid will leak out of the one end. That's it. Okay, now we will charge the Nuba chamber. First of all, put this cover slip over the Nuba chamber. Now make sure the outer edge of the cover slip must be slightly inside the outer edge of the Nuba chamber. After positioning the Nuba chamber, first of all, discard the first drop of the Nuba chamber, which is inside the stem portion, which does not contain any RBCs, so it is of no use. With the help of second drop, we will make contact of this second drop over the edge of the cover slip. Okay, so don't put this drop over the cover slip. It should be made contact slightly just side of the cover slip. Okay, as you can see, it will charge the Nuba chamber. It should not be undercharged or it should not be overcharged. Undercharge means the charging is done slightly less than this Nuba chamber. Overcharging means fluid goes into the gutter of this side edge, that is edge-shaped gutter. So that is overcharging. So it should not be undercharged, it should not be overcharged, it should be optimum charge. Okay, after optimum charging, put it for the 50 seconds or a minute. After wait of minute, put it under the microscope, which is already focused. Okay, and switch on the microscope and start counting. That's it.
Now because in the video it is difficult to show you the charging, these are the reference photographs for you. The first one is a photograph of a undercharged chamber. As you can see, the charging is insufficient to cover the whole Nuba chamber of the one side. In the middle photograph, it is optimally charged. As you can see, the one side of the Nuba chamber is fully charged. And in the third photograph, as you can see, the fluid is overflowing out of this right side of the Nuba chamber to the left side and in the central depressed area or gutter. Okay, so the right one figure is of overcharge, left one is a undercharge, and the middle one is a optimally charged. So it should be optimally charged. Okay, now counting the cells. Now as you can see, Nuba chamber will look like this. Now in Nuba chamber there are 9 squares. You have counted WBC in 4 corner squares previously. Okay, written with the WBC. Today you have to count RBCs in the central square. Now as you can see, in the central square there are further smaller 25 squares. You don't have to count RBC in each and every 25 squares. You have to count RBCs only in 5 squares written with the R, red R. So in those 5 squares you have to count RBCs. Okay, smaller 5 squares out of the 25 squares. That's it. Now the smaller square which will look like this. As you can see, in the smaller square there are further 16 squares are there. Now such 5 squares will contain 80 squares in which you have to count RBCs. Now let's see some dimensions of the Nuba chamber. As we know, Nuba chamber has a dimension of 3 mm by 3 mm. So the each square has a dimension of 1 mm by 1 mm. Now further square it down, the smaller square will have dimension of 0.2 mm by 0.2 mm, that is 1 by 5 mm by 1 by 5 mm. Now that will be length and width, and depth will be the space between the cover slip and Nuba chamber. As you can see in the marker, this space will be 0.1 mm, that is 1 by 10 mm. So that will be the depth. So this dimension will be considered when we will calculate the volume of each square. Okay, so length, width and depth. Depth is a space between the cover slip and Nuba chamber. Okay, so that is all about the dimension. As you can see, this is the smallest square of the 16 square. While counting, you have to follow some rules. Now as you can see, RBC will look like this, transparent bag like structures. Okay, there will be no color to the RBC because we have not used stain. So RBC will look like this. Now you have to count those RBCs. Now you have to follow the rules, same rules that you have followed in WBC count. So again I will revise it. Now as you can see, these are the RBCs. Now the RBCs which are fully inside the square, you have to count them definitely. So these 6 RBCs will be counted. Now apart from that, the RBCs which are over the sides of the square, that is over the borders, all of them you don't have to count. You have to count only over the left side and lower side. So these 4 RBC will be counted, but upper side and right side you have to ignore. So these 2 RBC, red colored RBC, will be ignored. Okay, so will not be counted. Now some RBC will be over the corners, like the 4 remaining RBCs. Now out of these 4, you have to count only left lower corner one. So the single RBC will be counted out of these 4, and the other 3 will not be counted. Okay, so now collectively the green ones will be counted and the red ones will not be counted. Okay, so these are the rules that you have to follow while counting the RBCs. Okay, so these are the same rules that you have followed in WBC count as well. So here you have to follow the same rules. Okay, so that is all about the some rules, tricks about the counting the RBCs. Now after you have counted the RBC, we have to do some calculations, because what we have counted will be in the diluted fluid. So now the calculation portion that we have done already in WBC count as well. So here also we will do the calculation. In calculation there are 3 steps. First step will be the calculation of the volume. First of all, calculation of the volume of one square, that is length, width and depth. As we have discussed earlier, the length and width will be 1 by 5 mm by 1 by 5 mm for the single square, and the depth will be the 1 by 10 mm. So collectively volume of one square will be 1 by 250 mm cube. So volume of 5 squares in which we are counting the RBCs will be 1 by 50 mm cube. Second step will be the calculation of the dilution factor. Now we have collected the blood up to 0.5 unit and we have diluted up to 101 unit. But as you can see, the part of the fluid in the stem portion, that is one part, will not take part in the dilution. So the final dilution will be 100 parts. So 0.5 part in 100, so 1 part in 200. So the final dilution factor will be 200. So we have to multiply the figure with the 200. Now the last step, that is step 3, total RBC count. Now we have counted the RBCs in 5 squares, but we have to make a unit of 1 mm cube. So we have to convert 1 by 50 mm cube into 1 mm cube. Now we have counted, suppose, n cells in 5 squares. So n squares in 1 by 50 mm cube, so how many cells in 1 mm cube? That's it. And we have to multiply it with the 200, that is dilution factor. So the ultimate calculation will be n into dilution factor upon the volume of 5 squares. So ultimately n multiplied by 10,000 cells per mm of cube, where the n is total numbers of cells counted in 5 squares. Okay, so that is your final calculation. Okay, now about the last part, and that is disadvantages or sources of errors which are present in this method. As we have discussed earlier, RBC count is not preferred nowadays in clinical practice because of the errors which are present in this method. But now automated methods are available, so this method is almost obsolete. In this method there are chances of 15 to 30% of errors. Now these errors can be either inherent, which can be visual errors, or errors due to the distribution of the cells. Sometimes cells will not get distributed equally in all these squares of the Nuba chamber. So in some squares you will count more cells, while in some squares there will be a lesser distribution of the cells. So because of that some errors may occur. So those are inherent methods, inherent errors in this method, but some errors may be due to the human errors. So one is dilution error. Some may not dilute fully up to 101, so dilution may not occur up to the mark, so because of that count may increase. Then the pipetting error, blood which should be taken up to 0.5 may not be taken up to 0.5 or increase or decrease. Then chamber error, sometimes some chambers are duplicate one, so those may cause problems. Then charging error, charging must be optimum, not overcharging or undercharging must be done, so that may cause error. Then counting errors, as I have told you, you have to follow the rules of L, left and lower, so those rules may not be followed, so that may cause counting error. And then finally calculation error. So these are the some errors which may occur in this manual method. Okay, so that's why this manual method is not preferable in clinical practice. Okay, so that's it about the RBC count. Thank you.